Hey everyone! As we start to explore the digestive system, we're going to conduct an experiment to test the types of macro and micronutrients found in food. Food is made up of important molecules that our bodies and our cells need. Our digestive system does the work of breaking down our food into the smallest molecules so they can be made available to our cells. Today we will be testing an apple, some cheese, and a cracker. These are foods you may choose to eat either for snack or for a meal, and you might even have some ideas as to why these would be good picks for your body's nutrient needs. Before we dive in, take a minute and think about what nutrients you might expect to find from these foods. The food we eat is made up of three main types of molecules: carbohydrates, fats, and proteins. These molecules are too large for our cells, so the digestive system breaks them down into smaller molecules. Carbohydrates break down into simple sugars like glucose. Fats break down into fatty acids. And proteins break down into amino acids. Each of these smaller molecules has an important role to play in the health of our bodies. Glucose molecules are used by cells to produce energy for the body. Fatty acids are used to help protect the nerve and brain cells. And amino acids are used to build new proteins in cells and for cell repair. First, we'll use iodine. Iodine is a brown liquid when this liquid is in the presence of starches. So starch is one macronutrient that we find in food. When there are starches present in the food, the iodine will actually turn to sort of a blue-purple color. So if any of these foods have starches in them, then when we use iodine with the food, we'll see it turn a blue-purple color. We are also going to use what's called a Benedict solution. So that's this one right here. It's this bright blue. Now this tests for simple sugars. Starches will break down into simple sugars. So simple sugars are actually our micronutrients. They're the smaller molecules. So this is going to find those smaller molecules. And when it is in the presence of those smaller molecules, if those are present in any of these items, it's going to turn to a yellow-orange color. And then finally, we'll use this solution here. It's called thyrate reactant. Now, thyrate reactant is going to test for proteins. So proteins are another common thing that we want to eat food for. That's a very important part of our nutrition is proteins. And so if there is proteins present in any of these items, then this is going to turn into a violet color. And so you can kind of see it's like a dark blue color, and it's going to change into more of a pale purple color. So we'll be able to take a look at that. Let's go ahead and start with a cracker because that's going to be a little bit easiest to work with. So we'll start with that. We'll put these two off to the side for now. The first thing we need to do is we need to turn this into a solution. So we can't just put these directly onto the food. It has to get into a solution. And as we're thinking about the digestive system as we do this, this is exactly what our digestive system would do as well. So think about how your food starts out. It starts out in your mouth, and your teeth go to work crushing it up, just like we're doing here. And then saliva is added to it, so it kind of becomes this mushy solution. And then as it gets added or gets swallowed, it travels down the esophagus into the stomach. And then the stomach adds more stomach acids to it, so that by the time it enters into the small intestines, it's actually a liquid solution. And that is where the nutrients can be extracted from. So let's start with the iodine. Now remember, iodine is testing for starch. And so if there's starch present in the cracker, so here's my cracker solution. You can see it's kind of that clear color. The iodine solution is this brown color. If there is starch present in this solution, in this cracker solution, then it's going to turn to a blue-purple color. So we'll go ahead and open it up and use our pipette to pull some out. All right, so we're going to add our iodine to our cracker solution. I'm just going to add a couple of drops. Don't want to add too much. And we're looking for a change. Oh, do you see that? Do you see? Oh, yeah, there we go. All right. Do you see how it's turned into a nice blue purple? It's a very dark blue color. That's actually really pretty. Yeah, so we can see a really nice dark blue color there. All right, that looks really good. So now with our second one, let's test that one. I'm going to put this off to the side. And let's test that one with our Benedict solution. So remember, this is going to test for simple sugars. So in the digestive system, these carbohydrates, these starches, will actually break down to the simple sugars because the simple sugars are one of the things our cells need. So sometimes food already has the simple sugars, and then the digestive system has less work to do. And that's like why the simple sugars are kind of a quick energy boost. But let's see if that's the case with our cracker. It should turn a yellow-orange if it has simple sugars. And it doesn't seem to be showing a change, which is kind of to be expected since it was made up of since we saw that it was made up of the carbohydrates of the starches, and we know that starches break down into sugars. The carbohydrates, the starches are complex molecules. The stomach, the intestines are going to do the work of breaking it down later on. So it makes sense that there's no simple sugars in there.
Sometimes it's helpful to compare the results to help observe changes. Using iodine to test for starches, we can see that the crackers showed positive results, while the apple showed negative results, and the cheese also showed negative results. We used the Benedict solution to test for simple sugars. This showed that the cracker had negative results, the apple had positive results, and the cheese had negative results. The birate reagent was used to test for proteins. This showed us that the cracker had negative results, the apple had negative results, but the cheese had positive results. Testing for simple sugars using Benedict's solution in fruit is a little tricky. This is because Benedict's solution will change from a yellow-green, almost a lime yellow color like what we saw with the apple, when the level of simple sugars is low, and to a bright orange-red when the level is high. Fruits are made up with a variety of sugars, including fructose, glucose, and more complex starches, depending on the fruit. You can expand on this experiment further by testing out other fruits and even other sweet treats like candy with the Benedict solution. Fruits and other things with higher levels of simple sugars will produce a brighter orange-red color during the reaction, possibly taste sweeter, and they also break down quicker in your digestive system, giving you a quick burst of energy that may need to be replenished sooner. Take a look at this comparison of the apple with tests conducted on a banana. What do you notice? What does this tell you about the nutrients of the banana? Is it made up of the same macro and micronutrients? What are some other foods you might consider testing as you continue to explore the macro and micronutrients that make up the food that you eat?